Hello, we're glad you've joined us for this live webinar, Surveying Expression of Immune Checkpoint Markers in the Tumor Microenvironment. I am Judy O'Rourke of LabRoots, and I'll be moderating this session. Today's educational web seminar is presented by LabRoots, the leading scientific social networking website and provider of virtual events and webinars advancing scientific collaboration and learning. It's brought to you by Advanced Cell Diagnostics. Advanced Cell Diagnostics is a world leader in RNA biomarker analysis for precision medicine, providing the most advanced RNA analysis solutions for cell and tissue-based diagnostics in molecular pathology. Its proprietary award-winning RNAscope technology is capable of detecting and quantifying RNA biomarkers with single molecule sensitivity and single cell resolution. The technology has been employed in hundreds of peer-reviewed publications by a rapidly growing customer base of thousands of active users, including all the top global biopharmaceutical companies and world-leading academic institutions. The company builds strategic alliances with its customers and partners to develop companion diagnostic tests for existing and emerging therapeutics, and also pursues internal programs to develop and commercialize proprietary cancer diagnostic tests. To learn more, visit www.acdbio.com. Let's get started. This webinar has been approved for continuing educational credit. Please click on the CE button at the bottom left corner and follow the process to receive your credit. You can post questions to the speaker during the presentation while they're fresh in your mind. To do so, simply type them into the Q&A box, which will open when you click the green Q&A button at the lower left of the presentation window. Questions will be answered after the presentation. To enlarge the slide window, click on the screen icon in the lower right-hand corner of the slide window. If you experience technical problems seeing or hearing the presentation, just click on the support button found at the top right of the presentation window, or report your problem by clicking on the green Q&A button at lower left. I now present today's speaker, Jeffrey Kim, PhD, a research scientist for R&D at Advanced Cell Diagnostics. Dr. Kim obtained a PhD in molecular biology at the University of Connecticut, investigating how nutrient metabolism impacts gene expression to alter chronic disease states. Afterward, he completed a postdoctoral fellowship at the University of California, Davis, looking at oncometabolite metabolism in renal cell carcinoma and the role of docosahexaenoic acid-derived metabolites on tumor angiogenesis. He previously completed a fellowship at the National Institutes of Health examining the influence of various fatty acids on brain development by combining the powerful tools of mass spectrometry and imaging. Dr. Kim will now begin his presentation.
Thanks, Judy. Hi, everyone. I want to thank you for joining me today as we discuss the expression of immune checkpoint markers in the context of the tumor microenvironment. By now, it comes as no surprise that immune cells have an influence in the development of cancer. Hence, immune monitoring is a valuable approach in assessing and discovering meaningful relationships and interactions between the host immune system and tumor. The immune system plays an important role in controlling and eradicating cancer. It's a personalized defense system that is specialized in recognizing and attacking anything identified as being foreign in the body, such as viruses, bacteria, and even the body's own mutated cells. While it is extremely effective at clearing out these potential harmful intruders, it's when cancer cells become overly mutated to the point that the immune system fails to recognize and kill them that they grow unchecked. In these cancer tissues, immune suppressive cytokines, molecules, and cells including Tregs constitute the immunosuppressive network to further inhibit effective anti-tumor immunity, thereby promoting cancer progression. This horrifically allows the tumor cells to generate a tolerant microenvironment and activate negative regulatory checkpoints. This should all sound quite familiar to the cancer immunoediting hypothesis put forward by Schreiber many years back. While this may all seem bleak, the blockade of immune checkpoint molecules have changed the landscape of the immune battlefield. As with many things, a careful balance is important. The case here, a balance between inhibitory checkpoints and activating signals, is necessary to regulate an effective immune response. Many cancers protect themselves from the immune system by inhibiting T-cell signaling, such as the circumstance of the inhibitory checkpoint signaling of programmed Death-1 receptor, also known as PD-1, by its ligand PD-L1. Effective therapies blocking PD-1 or PD-L1 may prevent the downregulation of T effector cell functions, allowing T cells to maintain and even regain their anti-tumor ability to mediate tumor cell death. What this means is that antibody therapy directed against these negative immunological regulators, these checkpoints, have demonstrated significant success and will likely be a major component in patient treatment for various malignancies. Clinical trials targeting PD-1 and PD-L1 in melanoma and non-small cell lung cancer have reported high response rates, and tumoral PD-L1 expression has been suggested as a potential biomarker to enrich for patient response to these treatments. Recent clinical trials utilizing PD-1 or PD-L1 antibodies in melanoma, non-small cell lung carcinoma, and renal cell carcinoma have shown safety and durable tumor regression. Anti-PD-1 antibodies have recently been approved for use in the United States and other countries. Anti-PD-1 therapeutic antibodies function through binding to PD-1 on tumor-reactive T cells, thereby inhibiting the PD-1-PD-L1 interaction, leading to reinvigorating the anti-tumor T cell response. PD-L1 is normally expressed on hematopoietic, endothelial, and epithelial cells in response to inflammation, and expressed on a number of tumors, including ovarian, lung, renal cell carcinomas, which are believed to play a major role in the immune suppression within the tumor microenvironment. Specifically, PD-L1 expression in tumor cells have been shown to suppress T cell activation and induce T cell apoptosis. From these various studies, we know that antibody therapies directed against these checkpoints fortunately work for many patients. However, why do we see varying levels of success? Anti-PD-1, PD-L1 drugs show consistent activity across various solid tumors. However, a substantial proportion of patients show a primary resistance, and those who benefit do not benefit to the same extent. Positive expression of PD-L1 through immunohistochemistry has been previously used as a screening tool. However, inconsistencies and reliability issues remain present. For example, some patients demonstrating PD-L1 positive tumors have no response after therapy. Alternatively, clinical efficacy has been demonstrated in PD-L1 negative tumors in other patients. So why do we see this variation using immunohistochemistry? It may fall on the use of nonspecific antibodies, inconsistent cutoffs, including mutable definitions for positivity, and tissue preparation and processing. In many antibody trials, simply looking at clinical outcomes rather than evaluating actual immune responses has made it difficult to know how to improve the approach. One of the many things one must consider are the antigens present. To accomplish this, a cell-specific and sensitive approach is indispensable to tease out these previously used response parameters that have been quite incongruent. In order to bring meaning to the data I will present to you today, allow me to provide a brief introduction to the technical approach so that we may appreciate the data and see how it can be utilized in furthering our understanding of immune checkpoint markers. First, let's discuss the RNAscope technology. The technology behind the RNAscope assay has three main advantages over conventional gene expression analysis methods. One, it generates very high signal amplification with suppression of background compared to traditional RNA-ish. Two, it can detect signal RNA molecules in single cells with morphological context, unlike random bind methods such as PCR. And three, it's universal. The assay can detect virtually any gene in any tissue from any species, provided the sequence is known, unlike immunohistochemistry, where getting an antibody to perform with specificity and high sensitivity is a very challenging prospect. Also, if an antibody is not available, it can be very time-consuming and costly to develop one. And now, here's a brief overview of the RNAscope method. It's a slide-based assay intended for use on the same types of samples you would use for either immunohistochemistry or immunofluorescence, either formalin-fixed, paraffin-embedded, or fresh or frozen, fixed frozen tissue sections or cells. The RNAscope methodology involves hybridizing the target-specific oligonucleotide probes to the target RNA. The target probe is a pool of oligos that we've designed using an established informatic algorithm that selects sequences to specifically bind physically to the target sequence and not cross-hybridize with any other sequences, yielding unparalleled specificity. The end result: a single target dot representing an RNA molecule. This visualization of punctate dots allows for quantification of gene expression. Because tissue samples can be highly variable, proper sample preparation and the right controls are essential to an RNAscope experiment. Tissue samples must be properly fixed and prepared so that there is a good quality RNA in the sample and no background due to poor fixation. ACD provides guidelines on how tissues should be fixed for optimal performance for the RNA assay. Here are serial sections from human lung cancer samples on which we performed the manual red chromogenic assay. On the left is the section stained with a negative control probe DACB, which is a bacterial gene that is not expressed in intact tissue. As observed in this sample, there should be no signal from DACB. In the middle panel is a section from the same sample stained with our positive control for the housekeeping gene PPIB. As observed from the sample, you want to see fairly uniform detection with a positive control probe indicating good quality RNA. Certain tissues may express the positive control probe at higher or lower levels, but in general, you want to see expression throughout the sample. With the results from these two probes, we can have confidence in the test data we see in the right panel. This is a section from the same lung cancer sample probed for PD-L1. Trust me, we'll see a lot more PD-L1 today. PD-L1 exhibits a wide range of expression in tumor tissues. In this human lung cancer sample, we observe strong punctate dots with PD-L1 probe in the sample. The controls show us that there is good quality RNA throughout the tissue sample and that there is little to no background. So we can be confident that the localized pattern of expression we observe for PD-L1 is in fact the true reflection of the biology of the tumor. And here is an image of a non-small cell lung carcinoma sample, which will be the focus of the study presented to you momentarily. Evident in the above image and throughout the study, we observed a great deal of intratumor heterogeneity of all markers in almost all tumor samples. Starting from the left-hand side, the expression of PD-L1 is expressed in most of the cells at a high amount. Going towards the center, we see that when we cross the middle divide across the stroma, PD-L1 expression is higher than the first region and slowly diminished further into the tumor. You'll also notice little pockets of PD-L1 that are also scattered throughout the tumor region. Non-small cell lung carcinoma is the leading cause of cancer-related mortality worldwide. While its treatment has been dramatically improved in patients who harbor targetable genomic alterations, such as EGFR, only a minority of small non-small cell lung carcinoma patients benefit from these targeted agents. Immunotherapy approaches, such as PD-1 and PD-L1 blockade, appear to be broadly efficacious in non-small cell lung carcinoma patients. For this immuno-oncology application, we at ACD's R&D team conducted a study on non-small cell lung carcinoma to evaluate tissue expression of immune checkpoint markers and immune functions in the tumor microenvironment. We selected 60 FFP tissue blocks from non-small cell lung cancer patients. These 60 tissue blocks were already screened and QC passed for RNA quality by using the positive control probe PPIB and the negative control probe DACB. Out of the 60 cores, 56 were tumor tissues and 4 were found to be adjacent non-tumor tissues. Out of those 56 tumor cases, pathological evaluation indicated that 26 cases were squamous cell carcinoma, 22 were adenocarcinoma, and 8 were a combination. The 60 tissue samples were arranged as a tumor microarray on 2 slides as depicted in the schematic illustration. Here, yellow circles indicate tumor tissues, while white circles indicate adjacent non-tumor tissues. We first evaluated the PD-1 gene expression profile by using the RNAscope singleplex assay, followed by evaluating co-expression profiles of PD-L1 with one other checkpoint marker or immune functional marker in the tumor microenvironment by using the RNAscope duplex assay. Here is an overview of PD-L1 expression in selected TMA cores. PD-L1 mRNA is shown here as red dots. In some of the TMA cores, such as the no PD-L1 example, black depositions of anthracotic pigment were present, which is an accumulation of carbon acquired from the environment. These dark deposits should not be confused with the RNA-specific signal. In these selected cores, a dynamic range of PD-L1 gene expression is visualized, from high, medium, low, to no PD-L1 expression in the various tumor cores. In some adjacent non-tumor cores, we observe rare PD-L1 positive cells, likely representing antigen-presenting cells. The negative control DAPI probe in serial sections of the same tissue show no background across all sections. RNAscope data can be analyzed by both a semi-quantitative and quantitative method. With a semi-quantitative method, a score is assigned to a sample based on the average number of dots per cell. Shown here at the top is the semi-quantitative scoring system that ACD has generated for the assay. While a score of 0 has no staining or less than 1 dot for every 10 cells, a score of 4 has greater than 15 dots per cell. This method will be used to provide a general understanding of the results. But later on, we will look at data analyzed using an automated approach. But for now, on to the data. This again is a schematic overview of PD-L1 expression of all 60 cores. Numbers in each circle represent the scores generated by the manual counting, which was explained previously. Among the 56 tumor tissues, 82% were found to be PD-L1 positive, with scores of 1 or higher consistently for 2 independent experiments. 56% of the tumors showed medium to high levels of tumor PD-L1 expression, with scores of 2 or higher, which was also tested in 2 independent experiments. Overall, this study demonstrates a high percentage of PD-L1 positive tumors in this subset of non-small cell lung carcinoma patients. This number is higher than previously reported cases. This may be attributed by the fact that the RNAscope assay has high sensitivity compared to immunohistochemistry. Additionally, this can simply be a reflection of a unique environmental status in this subset of patients. Next, using the RNAscope duplex assay, we then evaluated the expression landscape of other checkpoint markers in combination with PD-L1 in serial sections of TMA. This table here shows the distribution of tumor tissues expressing these 8 additional markers, including immune inhibitory checkpoint markers such as PD-1, PD-L2, LAG3, CTLA4, TIM3, and B7H4, IL-10, a cytokine marker, and the immune stimulatory molecule 4-1BB, analyzed together with PD-L1. The expression pattern was represented by percentage of tissues with corresponding RNAscope scores. For example, 54% of tissues showed low PD-1 expression with scores of 1, while 52% of tissues showed high TIM3 expression with scores of 3. From this table, we see that many tumors expressed high levels of CTLA4 and TIM3, while IL-10 and PD-1 expression were relatively low in most tumor cores. I'll now show you the expression profiles of these various checkpoint markers in selected tissue cores shown in this table. These cores were ranked from high to low PD-L1 expression from top to bottom. In core 2E2, where PD-L1 is expressed at very high levels, we also observed high-level expression for PD-L2, LAG3, CTLA4, TIM3, and 4-1BB. In cores 1B1 and 2D1, where PD-L1 is expressed high with scores of 3, the co-expression profile of the other markers between the 2 cores were variable, especially with B7H4. In cores 1Z1 and 1A2, where moderate levels of PD-L1 was expressed with scores of 2, variations of other markers between cores were also observed, especially with LAG3. And finally, in cores 1B3, 1F3, 1B5, and 2C3, where little to no PD-L1 was expressed, markers are found to be expressed in a heterogeneous manner. Overall, what this tells us is that there are complex and heterogeneous co-expression profiles of various checkpoint markers in the tumor. The information presented here brings potential interesting insights on therapeutic approaches. For example, with tumors such as 2E2 and core 1B1, which show high levels of PD-L1 and CTLA4, respond better with combination therapy, or with tumors like 1B3 and 2C3 still be responsive to other checkpoint inhibitors, even though little to no PD-L1 is expressed. Since each tumor is clearly exhibiting varying expression profiles of these markers, it goes without saying that each patient could experience different successes if given the same therapy. Here we have a snapshot of core 1B3, which shows low to no PD-L1 expression. Since these TMAs were serial sectioned, similar regions can be compared side by side. We find here that there's little co-expression of PD-L1 with PD-1, PD-L2, and 4-1BB in these selected regions, while moderate expression of LAG3 and TIM3, and finally strong CTLA4 expression was found in the stromal regions. In contrast, we have co-expression snapshot of core 2E2 here, which showed high overall PD-L1 expression. From this serial sectioned tumor region, we observe that PD-L1 is shown in teal, while all other markers are shown in red. We find that PD-L1 and PD-L2 are both expressed in tumor and stromal regions and co-expressed in the same cells, which are indicated by the black arrows. CTLA4, 4-1BB, PD-1, LAG3, and TIM3 were expressed primarily in stromal regions. And lastly, LAG3 and TIM3 are co-expressed with PD-L1 in various cells. The following slides are zoomed-in images of each co-expression profile from the same core 2E2. Here again, we have co-expression of PD-L1 with PD-1, where we see PD-L1 expressed primarily in tumor cells, indicated in teal, and PD-1 expressed in lymphocytes, passing through the stromal regions in red. Next, we show co-expression of PD-L1 and PD-L2, where we see both PD-1 ligands expressed primarily in tumor cells. Here, we have co-expression of PD-L1 with CTLA4, where we see PD-L1 expressed primarily in tumor cells and CTLA4 on lymphocytes in the stroma. Similarly, co-expression of PD-L1 and LAG3 observed here, where we see PD-L1 expression again primarily in tumor cells, while LAG3 is found in the lymphocytes, again in the stromal region. And finally, we observe co-expression of PD-L1 and TIM3, where we see PD-L1 expression predominantly in tumor cells and TIM3 expression on lymphocytes. Co-expression is again observed in various cells. Infiltration by lymphocytes into the tumor region is well depicted by TIM3 expression. From these co-expression profiles, it is quite clear that tumors are again heterogeneous and that no two tumors are identical. In comparison between these two tumor tissues, LAG3, which is expressed in both tumor and stromal regions in core 1B1, but primarily in the stromal compartment in core 2E2. TIM3, however, was expressed almost mutually exclusively with PD-L1 in core 1B1, but co-expressed in various cells in core 2E2. PDL2 was primarily expressed in stroma and core 1B1, but also expressed in both stromal and tumor regions in core 2E2. Moving forward, next I want to show you another way of analyzing the data. If the investigation requires a quantitative component to draw out additional conclusions, then the following will be of interest. RNAscope data can be analyzed in a more objective and rapid manner using an automated software analysis platform such as Indica Labs' Halo software. In order to speed up the process of counting the number of dots or events in each cell, Halo was used to assign regions, set up parameters, and process the data. This will all make more sense in just a minute. For the analysis of the inhibitory checkpoint markers, PDL1 and LAG3, we designated two different regions of interest. These included the stromal and the tumor regions. Additionally, the entire core was also processed so that a comparison to the remainder of the tissue core could be demonstrated. These regions here are identified as layers, where yellow areas represent stromal regions, green areas designate tumor regions, and the entire core within the red perimeter surrounding the cell. And here are the results. This analysis demonstrates that LAG3 and core 1B1 was found primarily in the stromal region, while the tumor region showed a lower expression. PDL1 reflected an inverse relationship, which is biologically sound as PDL1 is expressed primarily on the surface of tumors. The strength of this approach is that region-specific target expression can be identified as well as counted. One point that I would like to mention is that the quantitation of LAG3 and PDL1 from the overall core, again here in red, showed that both checkpoint markers were expressed at equal levels. However, it is clear from this analysis that this is not the case, as LAG3 expression was higher in the stromal regions and PDL1 expression was found to be higher in the tumor regions. Having the capability to analyze samples along with morphological context permits such interpretation, which is not made possible with the typical grind and bind assays, which mask the site of expression. Now, how about if you want to compare two different cores? Well, for the analysis of these two cores, we use the same designated regions, the stromal and tumor regions, as well as the entire core. Based off of our previous manual count, core 1B1 was found to have a high expression of PDL1, while core 2D1 had lower expression. This translated to our quantitative analysis here, producing a meaningful checkpoint expression profile output. While LAG3 was expressed primarily in the stromal compartments, with a reduction in the tumor regions, it is quite obvious that a higher number of cells in the stromal region expressed LAG3 and core 1B1 compared to core 2D1. In a similar vein, PDL1 was highest in the tumor regions for both cores. While LAG3 was expressed primarily on lymphocytes, we see the expression in tumor regions indicating the presence of lymphocytes infiltrating into the tumor regions. The strength of this analysis is that the expression of different markers can be compared directly between two cores, which represents biopsy samples. From this, it is evident that co-expression is an occurrence that may influence the outcome of checkpoint blockade therapy. And that co-expression is an important aspect of determining the applicability of immune checkpoint therapy. Despite cores 1B1 and 2E2 expressing a similar quantity of PDL1 as well as TIM3 and LAG3, the strength of this RNAscope assay can be appreciated. Here we compare co-expression between the two cores, with core 2E2 having a greater number of PDL1 and PDL2 co-expression. Even within the same core, co-expression can be quite heterogeneous. In the schematic diagram to the top left-hand side, we see PD-1 and TIM-3 expressed on two different lymphocytes. Alternatively, both inhibitory checkpoint molecules can be expressed on a single cell. The image below reflects this occurrence. PD-1 in teal is expressed on various cells, as does TIM-3 in red. However, we observe co-expression in several instances indicated by the arrows. A recent publication revealed this aspect in targeting specific immune checkpoints. In the case of lung tumors, adaptive resistance to anti-PD-1 treatment developed, resulting in the upregulation of TIM-3 in PD-1 antibody-bound T cells. What this suggests is that the upregulation of TIM-3 and other immune checkpoints may be targetable biomarkers associated with adaptive resistance to PD-1 blockade. The takeaway from this revelation is that the immune checkpoint landscape isn't as cut and dry, and drug resistance must be considered during therapy. In order to evaluate the appropriate approach regarding immunotherapy, the co-expression of multiple immune checkpoints in the tumor microenvironment must be assessed for an effective means of attack. To summarize the study, the expression profiles of immune checkpoint and functional markers were evaluated in tumor microenvironment of 60 NSCLC FFPE tissues by RNAscope. A large proportion of non-small cell lung cancer cases were identified as PD-L1 positive by RNAscope scoring, either in tumor cells and/or stromal cells. Over 50% of cases showed PD-L1 expression in both tumor and stromal cells. The detection of co-expression profiles of multiple checkpoint markers in the tumor microenvironment revealed a heterogeneous pattern of expression in different tumor tissues as well as intratumor heterogeneity. This information may reveal potential insight into combination therapies targeted against different checkpoint pathways. The bottom line from this investigation is that there's a great need for more biomarkers to be tested in combination to gauge patient response. And with that, I would like to discuss a little bit about ACD. ACD is a biotech company with a goal in mind to facilitate various arms of research, whether it be in the diagnostic or clinical setting or even the academic realm. The idea is that our technology will be able to enable researchers to make progress in their research goals. And with that, I would like to turn it back to Judy.
Well, thank you, Dr. Kim, for your presentation. A quick reminder for our audience on how to submit questions. Simply type them in the Q&A box found by clicking on the green Q&A button at the lower left of the presentation window. Dr. Kim will answer as many questions as time permits. And the first question is, with diversity in the tumor cells, how can we make sure that TMA can reflect all phenomena in the tumor nodule?
You could say it one more time?
Sure. With diversity in the tumor cells, how can we make sure that TMA can reflect all phenomena in the tumor nodule?
That's a good question. I think I'd start off by answering, you can't, of course, encompass everything at once. So you have to have specific markers that you're interested in to identify, I guess, your target that you're mainly interested in. So that's a kind of a vague or a broad explanation, but if you have a specific marker in mind or specific cell type or functional marker in mind, you need to use that marker and probe it together to see if there's co-expression.
Would it be possible to check both RNAscope and IHC staining on samples from same patient to evaluate the correlation, and has Advanced Cell Diagnostics done that?
Oh, that's another excellent question. So yes, we are currently in the works of comparing antibodies and using our technology at the same time. We haven't released that information yet because it's actually still ongoing. But that's a very important question that we get asked, I guess, in the past month, often many times. So coming soon.
Can multiple fluorescence dyes be applied in this ISH assay to detect markers once?
Oh yes, we also have a multiplex assay where you can combine, at the moment, with confidence, 3 different markers at once, and doing so would enable you to look at all 3 markers at the same time within the same region of the same tissue.
Is the variation of checkpoints tumor type specific or more likely individual specific?
Oh, sorry, Judy, could you say it one more time?
Sure. Is the variation of checkpoints tumor type specific or more likely individual specific?
Ooh, that question I cannot answer. I'd say both situations can be true. Yeah, that one, it's too hard to tell.
Please help compare this technology with classic immunohistochemistry tissue array, operation, time spending, or cost.
Sorry, Judy, could you say it one more time? I got a bad signal.
Absolutely. Please help compare this technology with classic immunohistochemistry tissue array, operation, time spending, or cost.
So a typical RNAscope assay, it depends on if you're doing a singleplex or duplex. A singleplex can take around 7 to 8 hours from start to finish, so it can be done in 1 day. And that gives you a lot of information. You have the morphological context of the tissue, as well as you can have all the various targets that you're interested in. And the beauty of this RNAscope platform is that you can have any target in mind. As long as you have the sequence, we can have a probe for you ready in 2 weeks, and we can guarantee that it will be specific and indicate or target the probe that you're most interested in.
Was image analysis of TMA scores done with, or cores, I'm sorry, was image analysis of TMA cores done with Halo software?
Yes, yep, and that was used with Indica Labs' Halo software.
How many markers can I stain at the same time for the same areas and cells?
So it depends on which assay you choose. If you're going with the traditional chromogenic assay, you can use two probes at the same time. However, if you want to use a fluorescent approach, then you can combine up to 3 at the moment.
And more questions may be coming in, but in the meantime, I would just like to ask you, I would like to thank you for your presentation, and I would like to ask if you have any final comments for us today.
This was a great opportunity to discuss this recent study that we here at ACD conducted, and I think the beauty of this is that it kind of reveals to researchers and anyone looking at or interested in looking at the tumor microenvironment in great detail. You can do various targets at the same time, so I think that has a lot of weight and can actually push forward the research significantly.
Well, thank you. I'd like to thank the audience for joining us today and for their interest and questions. And we would also like to thank our sponsor, Advanced Cell Diagnostics, for underwriting today's educational webcast. This webinar has been approved for continuing educational credits. Please click on the CE button at the bottom left corner and follow the process to receive your credits. This webcast can be viewed on demand through December 30th, 2016. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.